Good afternoon or good morning, everybody, depending on the time zone. My name is Mohammad Gholam Sabir, and I'm working with Dr. Albensi at Nova Southeastern University, Florida. Dr. Albensi requested me to host this journal club today, as he has to travel to Miami for an important meeting, but he'll be listening us. It's a great pleasure for me to introduce our speaker today, Dr. Andreazza. Dr. Andreazza is a professor in the departments of pharmacology and toxicology and psychiatry at University of Toronto, and holds a Tier 2 Canada Research Chair in molecular pharmacology of mood disorders, and also the Thomas C. Jacos Chair in mitochondrial research. Our research focuses on understanding of the role of mitochondrial function in mental illness, especially in mood disorders. And we are all in very good mood today. It's a sunny, bright, sunny day. And currently, Dr. Andreazza is evaluating the impact of mitochondrial dysfunction on neurotransmission using 3D organoids generated from induced pluripotent stem cells and from patients with bipolar disorder and or mitochondrial disease. Dr. Andreazza also founded the Mitochondrial Innovation Initiative, known as mito2i.ca, with the objective of uniting researchers from different medical fields with a common interest in unveiling the role of mitochondrial function and genetics in human diseases. As we all know, that altered mitochondrial functioning has been implicated in the pathophysiology of a wide spectrum of diseases, including neuropsychiatric disease like bipolar disorder. In today's lecture, Dr. Andreazza will present an overview of current understanding of the involvement of mitochondrial dysfunction in bipolar disorder, and she will also highlight the impact of mitochondrial function on disease trajectory, severity, cognitive function, comorbidities, and inflammatory markers. So please enjoy Dr. Andreazza's lecture and enrich.
And thank you very much for the kind introduction, and thank you for the invitation to be here with you today. I apologize if my voice got a little scratchy during the time. For some reason, I woke up with some sore throat this morning. Let's hope it's not COVID. So far, so good. Yeah, well, it's really a tremendous pleasure to be here. I understand that your group focus a lot on mitochondria as well with Dr. Albensi works, and especially in Alzheimer's. I will not be touching a lot on the Alzheimer's field today. We do have an interesting paper on the go for actually patients with bipolar disorder and their the risk of development of Alzheimer's because of the connection with mitochondrial dysfunction. But there is there is a background. If it is possible, mute yourself.
I'm requesting everybody to switch off their speaker, please. Sorry about that. Yeah, go ahead.
No worries. But since that project is not yet complete, I will probably refrain to present another time if you wish to invite me to come on another day. Today, I want to talk with you about the powerhouse of our cells, which you all know as the mitochondria. I'm sure I don't need to go very deep details of this because you are a group that absolutely understand that. But just to remind us that when mitochondria is dysfunctional, there is not enough energy or signaling or calcium buffer or many others function that the mitochondria control that will alter not only brain function, but muscles, cardiovascular disease. And as was said in the beginning of this talk, is also mitochondria is very relevant in many different diseases, including those associated with the brain alterations. So it will be important to understand the role of mitochondria in health and in disease to try to start understanding how to treat better patients that are suffering either of a primary mitochondrial disease or a secondary mitochondrial dysfunction. Mitochondria is a very interesting organelle which we have in our bodies containing proteins that comes from the nuclear encoded mitochondrial genes and from the mitochondrial DNA. I like this figure for some reasons, but the most for me interesting is that if you look at the colors of the electron transport chain and our respiratory chain and follow the colors of the dots in these lines, you can see that, for example, complex 2 is actually only organized by proteins that come from the nuclear encoded genes. So translation, transcription, and dynamics also controlled by a lot of proteins that comes from the genes that are encoded by the nuclears. Mitochondrial DNA forms proteins that are present in the complex 1, 3, 4, and 5. This respiratory chain is essential to maintain our cells breathing and producing energy, and the differential of the potential between protons and electrons, which forms this kind of notion of a battery that we have about our mitochondria. It's essential to be understood in more details in the brain. And the brain, most of the times, weight about 2% of our body weight, but uses actually 20% of that energy battery. And I want to pause a moment here and just invite you to go to the YouTube channel for Mito2I, and you will see a 90-second video that you will explain this entire lecture in 9 seconds. So if you have some time, feel free to go in mito2i.ca and or in our YouTube channel, and you will find a 9-second video that will give you a very nice little overview of brain and mitochondria. The mitochondria is essential for neurotransmission, and I'm sure you are aware of this. And one of the reasons is because the neuronal connections require high-density mitochondria. When this happens, the signaling between cell to cell forms very well, and the neurosignaling is well maintained in the brain. But once mitochondria becomes either damaged or decreased in the amount of mitochondria present in the synapses, the cells becomes to actually decrease the neurosignaling, and there is a problem on cellular communication in brain cells. As I was said in the introduction, my research focuses a lot in bipolar disorder. For those that are not familiar with this disease, bipolar disorders as a mood disorders, which are changes in, there are dramatic changes. So it's not being sad or happy, it's being euphoria, which is also mania, and depression, which is often associated with attempted of suicides in patients with bipolar disorder. For many years, we have been trying to understand the role of mitochondria in the energy production in bipolar disorder. Tada Fumikato, professor at the Riken Institute in Japan, has started this field by looking into mitochondrial DNA alterations in patients with bipolar disorder. And he have demonstrated that patients with bipolar disorder have increased levels of mitochondrial DNA mutation. Our group more recently has demonstrated that mitochondrial DNA content and mitochondrial DNA oxidation is also playing a role in postmortem brain from patients with bipolar disorder. In this graph, for example, you can see that in the hippocampus, we have high levels actually of mitochondrial DNA content, but is not associated with other brain regions. We also demonstrated in this paper that there are increased levels of mitochondrial DNA oxidation in the same region. Genes are also altered in patients with bipolar disorder, and that the graph that is here is actually the reason why I moved from Brazil to Canada. Professor Trevor Young, back in 2006, was studying the gene expression in patients with bipolar disorder. He conducted a microarray study with a non-target hypothesis, and to his surprise, the genes of mitochondrial electron transport chain was downregulated in patients with bipolar disorder, mainly the NDUF7 in complex 1 and COX6 in complex 4. I wrote an email back in that time to him and said I was interested in following up this study, and which was part of my master degree, which is the DNA damage in bipolar disorder. We identified the patients with bipolar disorder had high levels of DNA fragmentation in peripheral cells and leukocytes. And also during my my master's degree, I demonstrated that I was increased levels of antioxidant enzymes and the S100B in serum in patients with bipolar disorder. As part of my PhD, I moved to understand more target information in mitochondria function in patients in bipolar disorder, and trying to figure out ways of evaluating that in the peripheral samples and post-mortem brain samples from bipolar disorder. By the end of my PhD, I moved to Vancouver. At the time, Trevor was in Vancouver, and Professor Young gave me the opportunity to work with the BA10 area from post-mortem brain patients, samples from patients with bipolar disorder. We then did a filtration analysis and extracted the mitochondria fraction and the synaptosoma fractions, and evaluate the protein content and the levels of activity of complex 1 to 5. For our not surprise, we did find that the NEGF7 was decreased in patients with a bipolar disorder, which led to a very interesting area of investigation in patients with a bipolar disorder, mainly being that the patients with bipolar disorder now have decreased levels of the gene expression of NEGF7, but also decreased levels of NEGF7. Collaborators of our groups have demonstrated that the decreased levels of NEGF7 is not present in the periphery samples of patients with bipolar disorder. So this seems to be an alteration in brain samples. Currently, we are trying to understand that using brain organoids, which I will talk later a little bit on that. I see there is some chat here. Oh, all right. If there is questions, and you feel free to unmute yourself and ask me during the time. I'm more than happy to stop and answer your questions. In patients with bipolar disorder, it is also well known the role of metabolites, mitochondrial metabolites. In periphery samples, we are able to demonstrate that patients with bipolar disorder have high levels of lactate. And we several studies have demonstrated the patient with a bipolar disorder have high levels of lactate in the brain via MRS studies. This is interesting for us because lactate is a clinical relevant marker and could be actually well used especially in trials, to understand which patients will be benefiting of antioxidant treatments or not. There are several groups now around the world that actually are using this lactate marker as a secondary marker for patients with bipolar disorder to understand better their health. So in terms of treatment, this actually led to many different groups to start targeting alterations in the mitochondria and trying to find better treatments for this population. An example of a study is this is just unpublished data from the lab that is in the beginning of the studies. So, for example, we can take white blood cells from patients, transform it to induce pluripotent stem cells, and then generate neuroprogenitor stem cells. We took this as a complementary data for a clinical trial that we are developing with Professor Andy Nierenberg at Harvard School, where we're treating patients with bipolar disorder with bezafibrate, which you're probably aware is a mitochondrial biogenesis modulator acting on the PGC1 alpha. So we transformed in the cells from the patients into neuroprogenitor stem cells and treated them with bezafibrate. And as you can see in the figure A, the patients with bipolar disorder have decreased levels of ATP in the cells. And when you take the samples and look into the cell-free copy number, which is a marker of mitochondrial stress, the patients that had the control patients did not demonstrated a lot of an alteration. with this drug, one didn't react and the other, the copy numbers go up. So it's actually not beneficial for for control samples. But when you're looking in the samples from patients with bipolar disorder, the levels of stress of the mitochondria is high, and when treated with a bezafibrate, this goes up, the levels of stress go down. But one patient in this case and scenario did not benefit very much of that, absolutely in the very, very early stages of these studies, but an interesting to kind of see that is a responsive and is not equal to every patient. Back to the clinical trials, the bezafibrate trial is now almost closed. We are just finalizing the last patient's recruitment, and hopefully we'll see whether this medication is benefiting patients. This is an open label trial, so we did do a futility analysis by the middle, and I was able to see that 50% of the patients included in the trial did benefit of the bezafibrate treatment and decreased the MADRS, which is the depression scale levels for these patients, which is interesting that, again, that is not every patient that did benefit. Very similarly with the studies with N-acetylcysteine, which is its hopes is to increase the glutathione concentration in patients with bipolar disorder, improving their antioxidant capacity. In clinical trials using NSE, you see that that isn't a benefit of patients with bipolar disorder, mainly for depression states. However, secondary analysis of this and further clinical trials have demonstrated it's not as simple to treat patients with NAC, as some of them can actually precipitate mania in some of the patients. So further analysis is necessary to understand better. What a lot of research is focusing currently in the field of bipolar disorder is really to subtype the patients. We need good mitochondrial diagnostics to understand which patients with psychiatric conditions do have mitochondrial dysfunction and which don't. We do expect that those actually that have mitochondrial dysfunction will have an increased disease trajectory, will have an increased severity, a decreased cognitive function, increase of comorbidities. We are working tireless on the Mitochondria Innovation Initiative to actually provide good diagnostics for mitochondrial dysfunction in any patient suffering of any disease, but in this case will be very useful for patients with bipolar disorder to target therapeutics and novel treatments for these patients. One way that our group in patient-derived models. So we do work, and this is just an example of some of the types of families that my lab continue working. In the family one, what you see is a mother and a father which have not yet been diagnosed with any psychiatric condition or mitochondrial disease. However, there is a son with bipolar disorder and two deceased kids with cytochrome C oxidase deficiency. In family two, what you see is a mother with bipolar disorder, a daughter with no clinical psychiatric conditions or nor mitochondrial disease, but two deceased kids with cytochrome C oxidase deficiency. Important to note that further after we analyzed the genetic data of this family, was noted that the father actually had a mitochondrial disease called KSS, Kern's syndrome. And the family tree, for example, in this mother, she carried both mitochondrial disease from the cytochrome BC1 complex deficiency and bipolar disorder. And she has two very young child with a cytochrome C BC1. There is no psychiatric disease yet diagnosed in the kids. They are very young. They are under the age of 5. So these are examples of families that we've been working and towards the generation of brain organoids in these patients. We have published this method and is extremely detailed. So if you're interested in generating that in the lab, we hope that the methods that we have provided you in this paper actually will help you to achieve that in your laboratory. We do cellular characterizations for our organoids that goes from understanding whether the brain organoids have progenitor cells, neuronal cells, what stage of the cells. We do the neuronal activity, which will help us to characterize whether the presence and the activity of these neurons, if they are immature, mature, and in development. And in my laboratory, we do generate the iPSCs. Liliana Tiziano generates the organoids, and Dr. Martin Bulio does the electrophysiology. In my lab, we also look at the mitochondria function in these organoids, and we look in the different stages, in the white blood cells, in the iPSCs, and in the organoids. What we were able to demonstrate it with this is that we can, as you can appreciate, it's not a very simple process to get to the point of generating a brain organoid. It's 315 days to get to that point of an organoid that looks like that. In day 14 on the organoid generation, you have to start seeing the formation of these ventricles, and you want to see the kind of like a cloud shape on the organoids. When the organoids evaluated in this paper, particularly was 4.5 months old, and you should see radial glia coming in with SOX2. You should see postmitotic neurons, and you should see also presence of astrocytes marked here by GFAP. We then confirm that by measuring neuronal activity, and as you can see, the mature neurons marked in this graph by blue, they have a spontaneous activity, they have actual potentials, and they have a nice dip on your sodium current and potassium current. And we also note that in our brain organoids, we do have immature neurons and developing neurons, which was a good feature to be in that.
Sorry, the sound is kind of off now. Something actually happened.
Can you hear me okay? Or
yes,
no?
Yes, we are back now, yes.
Okay, awesome. Let me see if I just do this, maybe it will be better. Okay, so let me go back here. All right, so back we did then evaluated in these organoids. We track love mitochondria using MitoTracker, which is a dye, as you probably very familiar with, that only you only markers mitochondria that have mitochondrial membrane potential. We also confirm the mitochondrial membrane potential by doing a technique called JC1. And blocking the mitochondrial membrane potential by using FCCP. As you can see, the organoids generated from the iPSCs, they have mitochondrial membrane potential with the capabilities of being inhibited by a pharmacological inhibitor. That is oxidative phosphorylation. We did not show here, but is also on the paper if you're interested in reading. We did oligomycin treatment to understand how much the iPSCs or the organoids were actually using their ATP via oxyphosphorylation or glycolysis. It's noted that the iPSCs are more glycolytic, and the organoids are definitely start pushing more into the oxidative phosphorylation, which is expected to happen. We confirmed the morphology of the mitochondria with TEM. And as you can see in the iPSCs, the mitochondria are very different than the organoids, which also is expected to be that way. We hope these methods will lead us into personalize the treatment. We do not expect that we will have to develop brain organoids for every patient, but we do hope that these methods will help to characterize and understanding different types of bipolar disorder and many other brain diseases, and find the better treatments for each subgroup of patients. We study mitochondria, you probably know, is a big puzzle, and you need a lot of different information to come together. And that's why I founded the Mitochondria Innovation Initiative, and I invite you all to please join our website and become a member with our network. It's free, and you will benefit of access to videos, symposiums, and other activities that we promote. This network bring together patients, research clinicians, trainees, and fellows to working together to transform mitochondrial health. Our main goal is to take mitochondrial medicine from a niche field to become an everyday medicine. When you go to your doctor, we always try to measure our lipids and cholesterol to hopes that we will be preventing cardiovascular diseases associated with those alterations. Can we do the same for mitochondria? Can we start understanding better the function of mitochondria and preventing the disease associated with it, and target better treatments for them? I will stop here by thanking my lab, which are a fantastic group of students and staff, and thanking you for listening to me today.
Thank you, Dr. Andreazza. I would like the participants to ask any question. If you have, please.
Thank you.
Hello, thank you for a great presentation. Clearly, you're showing that inherited mutations in the mitochondria are part of the etiology of some of these patients. What about the development of somatic mutation over time? Is there an association with people who are susceptible and or aging, things like that?
Yeah, thank you for your question. There is in bipolar disorder, there is no good longitudinal studies yet. We actually just came together as a group of international researchers to establish the global bipolar cohort, which now is on year 2. So I think with that cohort, we will be able to start understanding better the effects of aging in this population, which is not clearly studied. There are some studies demonstrating that, for example, telomere length is decreasing in patients with bipolar disorder. We do know that patients with bipolar disorder die earlier than the rest of populations. There are more cancer prevalence in patients with bipolar disorder as well. But in terms of somatic mutations, I don't think I can recall a paper that have studied that yet, but it's definitely—
What would be an outcome of your oxidation studies? Greater oxidation would probably result in greater damage and therefore mutation. Absolutely. There's also a set of genes in the nuclear genome that have their effect in the mitochondria. They're required for proper mitochondrial function. Has anybody targeted them to see if similar effects can be seen?
You're talking about the nuclear encoded genes that promote proteins for— Yes, absolutely. There was many studies have been done, including studies in my lab. The one, for example, that you saw there was the NGF7, which is a nuclear encoded mitochondria protein, and that is translated from the nuclear genome and imported to the mitochondria. And this protein is decreased in post-mortem brain samples and decreased in also brain organoids from patients, but is not decreased in peripheral samples of patients with bipolar. So it seems to be a problem of importing of this protein from the nuclear to the cytosol, to the mitochondria on neuronal types of cells. Is also not shown in astrocytes. We don't find the alterations in NGF7 levels in astrocytes either from this patient population. So why? I think is still yet to be seen. We need more studies on importing mechanism. As you're probably very aware, importing mechanisms of mitochondria proteins are not fully understood. We just actually, the first paper of importing genes came like about maybe 3 years ago, not so long ago. So yeah, I think we will be able to see more coming. Genetic risk is also being studied in patients with bipolar disorder. A group from Mayo has demonstrated that there are about 20 genes that are nuclear encoded mitochondria genes that infer risk about 10% for 10% of patients with bipolar disorder. Sandra, nice to see you.
Well, if I could weigh in, first of all, I have to apologize. I'm a clinician scientist, as you know, and I had an emergency, so I missed your whole talk, but I got the end of it. I'm glad it's recorded, because I think you're right that we need to get this to start to be much more of our kind of standard, workup. But I wanted to comment first about the bipolar, because as you may know, Ben Goldstein, who's been very interested in this in youth bipolar, has demonstrated quite clearly that there is endothelial dysfunction that can be identified as a lack of cerebrovascular responsiveness to like breathing carbon dioxide and so on. In other words, they actually do not respond normally. And that can be partly reversed by physical exercise. And one of the reasons that, bipolar actually had a little role in getting Ben interested in this, because he was interested in it when he came from UPenn. And I commented on him as I don't see too many people with bipolar disease in my clinical practice and dementia. And that seems to be partly because they die of heart attacks. They die of suicide. They don't get to be in their 60s and 70s. And it's time to change that. Anyway, so it's very interesting that he's finding some biological biomarkers that are actually physiologically identifiable. But that's just a side issue. So I think we have to understand what mitochondrial disorders may have to do with that in the endothelial cells. But I did also want to say that this is a huge issue in Alzheimer's disease, right?
Yes.
So there is this idea that's type 3 diabetes, and there's not endothelial, there is mitochondrial dysfunction that's leading to problems in insulin signaling and glucose uptake, and that we have to attack this aspect of Alzheimer's disease as well. One tiny way we're doing that in an investigator-driven study is it turns out that amongst the ACE inhibitors and ARBs, perindopril and telmisartan are in each class, and they both penetrate the CNS much more so than the other ones. And perindopril has a little bit of effect on cholinergic function in the brain, but telmisartan there's animal work that shows that it accelerates the breakdown of toxic A beta in the cells and the astrocytes, wherever it's going on. And also it stimulates PPAR gamma, which seems to help with glucose uptake. So I think we're becoming more and more aware of mitochondrial function as part of what's going on in Alzheimer's disease. And then, as you know, also with the work that you do with Walter, very interested in white matter disease and the changes that go on in the small vessels of the brain, which again seem to be maybe identifiable through lipidomics in the blood. And I think you're very much part of what Walter is doing. So I think this is really starting to come on the radar, and investigators like yourself are hugely important to engage also the clinicians so that maybe we can start to get this to the front line. That was really just a comment I wanted to make. But I'm going to have to view your talk because I think it's been recorded.
Yes, it was recorded.
Thank you.
Yes, thank you very much for raising this point, Sandra. And it's quite a challenge, right? Because you want to make sure that we can actually take these biomarkers to the clinicians and ensure that we can actually measure in the patients. But at the same time, we want to make sure that we are measuring something that will be beneficial to understanding the disease and getting a signal of something. So I think we're getting there with the work that we've been closely doing with Ben. I think is partially we are closing to understand better how can we segregate the patients with the endothelial vascular disease and the biomarkers for mitochondria, how to treat them and how to diagnose them. So I think probably in the next 1 or 2 years, we should be able to see that coming into more clinical translation, which I think would be very beneficial. To your point about patients with bipolar disorder and Alzheimer's or Parkinson's, you are absolutely correct. It's a very small part of our population of bipolar disorder that achieve a long life. And for those that achieve, though, there is a higher prevalence of Parkinson's and Alzheimer's in that population, which is expected, right? If you see these alterations in the brain, with alterations in mitochondria, so you would expect that, but it's a very small portion of the patients that achieve over 60 years old, which we certainly do need to change that statistics.
Well, just to put that in context, and then I'll stop talking. The truth is we are moving towards blood tests, which are proteomics in Alzheimer's disease. And right now, as we speak, Quantarex actually has a chip that includes phosphatal 181, neurofilament light, and GFAP, and it's been FDA approved and it's marketed. And it allows you to know that a person has amyloid deposits in the brain because the phosphatal tells you that the amyloid is there. I think the amyloid measures in the blood are a little more tricky, and maybe mass spec does a better job because there's so many other amyloids floating around. But it's going to be important to not neglect the other metabolics. We're going to be in a proteomics fixation now for a while because that may be diagnostic. But it also means when you get blood, you can also get blood that will validly represent Alzheimer's disease, for example, pathology, as well as then be able to test people with some assurance that that might be the right diagnosis. Because we know that our clinical diagnoses without these biomarkers, and PET scans are very expensive right now at $5,000, for an amyloid PET scan. We make a lot of mistakes. So, 20, 30% of people don't have the disease we think. So that can bring precision medicine blood tests for proteomics to precision medicine understanding of the metabolomics, which means I think we're poised to be in a very interesting era. We should just be sending the blood samples to you guys as well. Okay.
Right. That would be lovely. Yes, absolutely.
So in this context, I really like this discussion, particularly Dr. Black's perspective on this matter. Like the endothelial dysfunction is a very important aspect of this whole spectrum of neurodegenerative diseases. But my question to Dr. Andreazza is that there are a lot of neuropsychiatric disease, for example, autism spectrum disease are actually neurodevelopmental, and there is some disease like Parkinson's and Alzheimer's, they are more like a neuropathological. So to what aspect do you think bipolar disorders fit within these two categories of disorders, neurodevelopmental and neuropathological? I would like your thoughts on that. And also, like this mitochondrial dysfunction is underlying a lot of disease, like almost every disease, there are some concepts that mitochondria is involved, starting from cancer to metabolic disorders. So my thoughts are that whether this mitochondrial dysfunction is a secondary effect, maybe the primary reason is probably something else, or because there are a lot of studies where people actually knock out different components of complex 1 or complex 4, and they actually have found different types of some unexpected physiological effect. For example, complex 1, if you delete, you see the dopamine signaling is not that much affected, and complex 2, if you remove the succinate dehydrogenase, then you see that there is some sort of paraganglioma, this kind of cancer. So your thoughts on that, like whether it is underlying all sorts of neurodegenerative disease or specific to particular kind of disease?
Thank you very much for those comments. Let me start with your first part when you were speaking about the neuropathology of bipolar disorder. I think for many years in psychiatric conditions, we have been diagnosed most of our patients through their behavior, right? We use the way the psychiatrist diagnoses by their clinical understanding and scales that measure the behavior of these patients. There is known yet markers of that are clinically used as biomarkers for bipolar disorder, nor to schizophrenia or depression. So I think that, in my view, has to change. And once we understand more of the neuropathophysiology of bipolar disorder, which currently involve alterations in the dopamine pathways, alterations in the glutamatergic pathways, as being both demonstrated to be part of bipolar disorder. Calcium signaling and mitochondrial dysfunction are the leading neuropathophysiology alterations in patients with bipolar disorder. We probably will understand better the disease, and we'll probably be able to segregate these patients. Perhaps we might not even be calling bipolar disorder, maybe we'll be having another name for the different biological alteration that this patient has. In terms of mitochondria, what we know is that about 25% of patients with mitochondrial disease, classical rare mitochondrial disease, will develop a psychiatric condition over time in life. And that meaning it's pretty high. And either it will be bipolar disorder or schizophrenia in this patient. So 25% is quite high out of this rare population. If you look at the opposite side, if you look for patients with bipolar disorder, how many of them have in the family someone with mitochondrial disease, you would think, well, that must be very rare. It's also not. It's also very present. It's about 10% of these patients, which is, again, striking to say, is mitochondrial dysfunction just a consequence? I probably will challenge you and say no. In some patients, some of the patients probably have an alteration on the pathways of mitochondria that is causing what we now characterize as bipolar disorder in these patients. There's alterations on the mitochondria that leads to alterations in neurochemical pathways and then in behavior that are then diagnosed as bipolar disorder. Is mitochondrial dysfunction a feature that is only for bipolar disorder? Absolutely not. I think as we more study over the times, we will define that there are different types of alterations in mitochondria that are related to brain functions. So we'll probably understand better that, as you said, alterations in the complex one might not be directly related with dopaminergic pathways, but we know that alterations in complex one change calcium buffering of mitochondria, and that is highly related with the release of neurotransmitters. And if you don't release properly, for example, serotonin, we know what can happen in the brain. You have what we call depression. So that's, I think, as we characterize more and more and understand that, we'll probably be able to have a branch of patients where we know that has a mitochondrial dysfunction and needed a treatment for that, and then the behavior will suppress because of that treatment. Is that going to be present in other diseases that are brain diseases? Probably yes. You probably have some patients with Alzheimer's that have mitochondrial dysfunction that could benefit of understanding better, and that will be necessary treatment. Similar will be for Parkinson's, similar will be for autism disease. So I think the future is bright, and I think what we're going to see is start like finding more the fine tune on how to treat and how to diagnose, most important. What I like to say is the who, how, and when of mitochondria. We need to know who has, when it started, and how to treat it. So that's the hopes of the mito triad that we have created, and hopefully we will see that coming more studies.
Right. Great, thanks. Also, in this context, because we have some pharmacy students joined here, I would like you to comment on what's the current therapy in bipolar disorder and how that therapy you think enhances mitochondrial function, or was there any research conducted tried to improve mitochondrial function to reverse the disease phenotype?
Yeah, so patients with bipolar disorder are mostly treated with mood stabilizers, lithium being the gold standard treatment for bipolar disorder. Some of patients do not respond well to lithium, then they will probably either go for lamotrigine, valproate, or other medications as well. Studies have consistently shown that lithium is actually increases mitochondria function, and I think there is some from your lab as well looking into that. I think Ida studied that as well on cells from animals, I believe, trying to improve. Valproate is actually quite the opposite, as a mitochondrial toxin. So if your patient has mitochondrial dysfunction as part of their bipolar disorder, you would like to avoid that. And lamotrigine and other mood stabilizers and antipsychotics have either non-effect or a minimal effect on benefiting mitochondria function. If you're interested on this topic, we recently published a paper with Tamas Kozik from Mayo Clinic on demonstrating which medications, which antipsychotics and mood stabilizers and antidepressants have an effect or not on mitochondria, and whether should or should not be prescribed to patients that have the mitochondria alterations. It's a very interesting read, especially if you're clinician and seeing patients. Yeah.
Can I bring up one other? So that's very interesting because there are anti-epileptics, and lithium is stabilizing and there are responders and non-responders, right? Which is very interesting. Yes. Hereditary, maybe different. But I've always been puzzled by MELAS, which is this recurring stroke and disappearing stroke, and it comes and it goes, and it's like, what is happening and why does it go away and then it comes again, and it sort of destroys people's lives. But one patient I'm following who's now in her 20s, and what's going on there? Yeah,
I hate to say that way, it's a fascinating disease in terms of the perspective of mitochondria. Is horrifying for the patients. So the stroke-like that you see in the patients with MELAS it's mostly created by a burst of energy production of mitochondria. So it's like the mitochondria stays dormant on the brain for so many times, and then it's suddenly inactivated and bursts too much energy, and end up leading to this seizure-likes, and then strokes right after. So Peter Carlin is actually, and Danielle Andrade, we joined forces on a grant we just finished writing yesterday on actually trying to unveil a little bit more of the role of mitochondria regulation and calcium waves and buffering into seizures and epilepsy and stroke-like events in MELAS patients. So not sure yet what we're going to find, but you are absolutely right. It's fascinating to understand why it just suddenly disappeared. Our hypothesis probably calcium related.
So that's actually important for me to know. This is like a little stage. Because there is a very interesting patient. Maybe I'll have Peter refer to Peter, because she's really, and she's being looked after in McMaster right now, but I'm not sure how long the person there is going to be active. So
I see.
She lives in Toronto, and it would be, I think she's very, very interesting.
Yeah, maybe Danielle Andrade as well could be another option.
Okay.
I think she's the main care for the MELAS patients currently. Okay. Yeah. Mm-hmm.
Okay. Yeah. So you have to come to a basic science group to find out who's the right person to go to. That's the problem with the University of Toronto. We're so big, we don't know about each other.
It's
So,
true.
so that is a very, you've just helped somebody, I think. So,
Awesome. That's
Okay.
fantastic.
thank you.
I will send you later an
I know.
paper on Mila's that
Yeah.
might be of
Great.
interest.
Great.
So I think that's great. I mean, I kind of particularly like the way this discussion is so productive and live. Also, like, since there is no other question coming, so I would like to ask another question. It's more like a perspective. So you are using pluripotent cells to develop this organoids model to study bipolar disease. So what do you think is the I mean, what are the limitations of different models, whether animal-based model or cell-based model, that can be helpful in studying this spectrum of disease?
Okay, you're talking about the iPSCs and the brain organoids, is that correct?
Yes. And
Yeah.
also, like, if you compare with mouse-based model, whether there is any good model to study bipolar disease in mouse, because mouse
No.
obviously do not talk like human.
Hugest problem in our field in bipolar disorder, there is no animal model for disease. It's very difficult to flip the animal from mania to depression. So how you would do in the mice is mania is characterized by hyperlocomotion, more hearing, so more exploring of the field, and the more sexual drive on the animals. And the depression would be characterized for, all the normal behaviors of being sleeping more, eating or drinking more or less than the animal is normalized, be retained in one single area, lack of exploration. So we can mimic either or in animal by one. For example, you can give amphetamine to them, that will be a pharmacological model, or you can block production of dopamine or block serotonin, and you cannot go either or. There is one person, her name is Colleen, that has got close to get an animal model for bipolar disorder using modulation of circadian rhythm, so genes. And she was able to go from a hypomania to depression. So like you get the animals walking a little bit more, and then they go back to a depressive state. We're not there.
So yes, so it's a big challenge, particularly. So what about the postmortem brain tissues? Like, is there a good repository for postmortem brain tissues from bipolar disease, and do you have access? Or?
Yeah, we do have the three main postmortem brain access that are now actually all conglomerate into the same biobanking at NIH. But a few years ago, there were three of them. So Harvard, Mayo, and I forgot the name, the Broad Institute. They had three. CAMAGE has a very small repository on that, but the three main ones are that. Australia has a postmortem brain bank as well, and actually a very good one, but they do not allow tissue to be used outside of Australia, which is a pity. Brazil has a repository as well, but also Brazilian laws does not allow the tissue to be shipped and analyzed. So the only clinicians and scientists local to the University of São Paulo will be able to actually.
Just a comment, I'm sure that the shock of the suicide makes it very hard for families to offer tissue, and it's coronary artery disease might be a better way to go, but there are a lot of ways to prevent that now too. Yes. So I don't know, it'd be hard to sort of get a consent in advance, say if anything does happen to me, I want to donate my tissue.
It's very interesting you said that, but McGill is doing that actually with the Gustavo Turek. They do have advanced directory with patients with psychiatric conditions, those that wish to donate their brains to science.
Maybe the people with familial disease might be willing to do that, right? As a sort of way to be studied and learned, but it's sort of I think when people actually die from suicide, it's just so hard.
Oh, it's horrifying. And that becomes a big problem for scientists as well. So for us, when we use postmortem brain, we like to have patients with a bipolar disorder that did not die via suicide, because the pathology of the patient that died via suicide is very different actually than the which is very, it is an interesting, the patients have a much more severe disease. They often have like a more, the depression is a longer period of times. The mania is less intense, is often just a hypomania. They have less times where they stay euthymic between the two. So they're very resistant to mood stabilizers. So it's a very different disease. I kind of feel that we would need to study separately the groups. Yeah.
That's great. Also, I'm learning a lot and probably will keep you asking more questions in future. Like one thing I also want you to discuss a little bit about this genetic predisposition to bipolar disease, because you showed some familial case history. Is there any genome-wide association? And we know that genome-wide association studies found a lot of SNP markers and other genes associated with almost every disease. But is there any genetic predisposition factor which you can do a screening in the population who are susceptible? Is there any test or this kind of whatever?
Quick answer will be no. A longer answer will be we do have genome-wide association. Studies has been performed in bipolar disorder. The most recent one showing 2 genes. One is the CACNAC, sorry. One is a CACNAC. It's a calcium channel gene, but it only present in a very small portion of the patients. So good, not so useful.
Strong, right.
The other one was a zinc finger protein that has been also demonstrated to be associated with the risk of bipolar disorder. Again, 1% to 2% of the population.
That's zinc finger is probably a transcription factor and one. Yeah. So
well, doesn't that bring up the possibility that basically we have the problem that we're not dealing with a disease, but a bunch of diseases? And one of the problems is going to be, can we split in a way that actually defines different mechanisms and allows us to approach treatment?
Oh, Stephen, if you could say that out of loud at every meeting you go, bipolar disorder is not one single disease, it's a bunch of diseases in one single pot, I would really appreciate that, because we've been, as scientists, we've been trying to push to stratification of this population. And not only for that, but a lot of the patients, for example, with depression, they might also be in a different category. We need biological tracing and segregation of this population, and stop looking just by the behavior to characterize our patients. Maybe from a non-psychiatrist, that's how I would wish that we would classify our patients. Yeah.
I mean, age of onset probably important, and also sex, I imagine, differences.
Yeah. You're absolutely right. In very early beginnings of my career, I studied with Professor Flávio Kapczynski, a phenomenal psychiatrist in Brazil. And one of my roles was mostly statistical analysis and modeling. And we noted that the age of onset of disease, but it was important, but more important than that was the number of years undiagnosed, because those years that they didn't have any treatment, the disease is speed up in severity higher. So that's another issue with any psychiatric patient, right? So they mostly wait until it becomes very severe.
Well, another way to,
sorry.
Oh, sorry.
Go ahead.
Just one of the ways to find different versions of this disease is to look in different populations, especially inbred populations. You might expect that in French Canadian population, you would get more of the same factors. and contrast that with Amish or Mormon. And basically that would allow you to define that there are things that go together and produce a syndrome that would allow you to create, bipolar 1, bipolar 2, etc.
You are absolutely correct. It would be fantastic. There is a group in Sardinia, in Italy, that actually study a very remote population, and that they have one of the most beautiful genetic data for psychiatric diseases. I don't remember if someone is doing on the French Canadian population. I would have to look back, but I'm assuming someone in Laval or the University of Montreal definitely probably looking at something that in that way. In Brazil, we do have a nice indigenous population on the south of Brazil that is very preserved area. And then there is a group from University of Pelotas in south of Brazil that it has done a longitudinal following of this population, including genetic assessment. And in their population, they have demonstrated some traces of that are very different to develop psychiatric conditions. So I do not remember the genes, but it's a pool of genes. It's a risk score genes. It's not a single gene, but very interesting actually studies on that.
I would have thought in Israel. I would have thought in Israel they must be all over.
Oh,
that.
Dorothy, you are yes. Thank you. Yes, absolutely. Dorothy Ben Sacker is the one that is doing loads of study on that as well.
Actually, we mostly think about Ashkenazi Jews, but there's actually 3 or 4 subgroups that also that have different segregating susceptibilities. So, the closer they are, but still distinct, allows you to look at, the commonalities and the differences. And
Sure. Yeah, absolutely right.
I have to run. I'm just delighted to have interacted with
I know.
everybody and to be able to put a few words in for people with different conditions. So thank you so much. I'm sorry I have to go now, but I will listen to your whole presentation.
Thank you.
And I appreciate the tip on the MELAS syndrome. So that's great. Thank you so much.
Thank you very much.
Thank you, Dr. Black, for joining us.
Thank you. My pleasure. Delighted. Bye.
Thanks.
It was wonderful to be here. Thank you very much for having me.
Thank you. Thank you. It was a very nice presentation and we learned a lot. So I end here and wish you all have a very nice day.
Awesome.
Thank you.
Bye now.
Bye now.